This is ComD 4000 Hearing Science. This is lecture slide set 12A, part 4. This covers study questions 756 through 765. This covers the slides in your slide sets from approximately slide 67 through 86. And then the very last few slides are what you have as slides 102 to 106. However, in the slides that you're going to be watching in this slide set, they were kind of taken a little bit out of order. Some of those middle slides were sort of put at the end to make this a more cohesive presentation. You'll see it as we go through, and I'll announce as we move through sort of where we are. This is your guest lecturer, Dr. Peter Ivory. And let's pick up then part 4 at slide number 67. So here we are at slide 67. This part 4 of 12A is going to talk about only really two basic things. It's going to talk about the time-intensity trading relationship that was discussed on slide number 27. And as well, we'll talk about the differential sensitivity for primarily for amplitude intensity and differential sensitivity for frequency, and talk a little bit about the implications of those. We're going to skip some other material that's in your chapter. We don't have study questions for those, so we'll just proceed with this abbreviated version of these main psychoacoustics points. So here we are at slide 67, and as you were told at slide 27, there's something called a time-amplitude trading relationship. What this is basically saying is that in order to maintain audibility of a signal, particularly at threshold, it has to have not only a particular amplitude, but it also has to have a particular duration. Again, we're talking about you select one frequency, so that's under control, and then you're adjusting the amplitude to find the least amount of amplitude. Well, the third dimension of any acoustic signal is its duration. And as it turns out, duration also has a factor to a certain point. And we can simply say it like this. There is a critical duration, which we'll see on the next couple slides, is 300 milliseconds. So at 300 milliseconds and above, additional changes in duration, additional increases in the duration of a stimulus above 300 milliseconds do not change audibility. Audibility will be constant as a function of duration above 300 milliseconds. However, as we'll see on a slide very clearly, a couple slides very clearly, once we start to reduce the duration of the test stimulus, when we're below 300 milliseconds and make it progressively shorter and shorter and shorter in duration, the person can only maintain audibility of that or will only be able to hear it if the amplitude is progressively made greater amplitude. So the shorter the duration, the greater the amplitude has to be in order to maintain audibility. That's the time-intensity trading relationship. You make one shorter, you have to make the other one longer. And fundamentally, that's what this slide 67 is basically saying, is that in order to maintain audibility of a stimulus at very short durations, you have to make it higher or greater amplitude. And that says the same thing. So the key factor here is that signals shorter than 300 milliseconds will be inaudible unless you increase the amplitude of that. Once we get to 300 milliseconds and longer, there's no difference in the audibility, and there's no relationship at that point. So this slide and another slide coming up talk about this, and it illustrates for us very clearly, as we can see here, that here's the threshold of audibility, and over here is the stimulus duration. And so somewhere at this elbow right there, we can see that we've got a change. And so from this duration on out, it's a flat line, meaning there's no change. So there's no change over here as a function of the change here. That point there is the point at which duration ceases to be a factor. But look what happens at durations below this. As we go for durations below this, in order to maintain audibility, you have to make progressively greater changes in amplitude. So for very, very short stimuli, we can still hear them, but we have to make them, approximately 25 decibels greater. And again, all other things being equal, this is the same stimulus. It's just very, very short here compared to over here. This particular phenomenon is known as the temporal integration time or the cochlear integration time. And it's approximately 300 milliseconds. So any presentation of a stimulus less than 300 milliseconds, we can only maintain audibility if you increase the amplitude. Once we get durations longer than 300 milliseconds, the duration doesn't have a factor. The clinical takeaway point on this, because this does have a direct clinical outcome, if when you're doing hearing testing, as you will do in COMD 4020, if the duration of the stimulus is shorter than 300 milliseconds, your person's not going to appear to hear that unless you make it, the next trial, you make it greater in amplitude. So you're going to make people look like they have a slight hearing problem when in fact you're just not leaving the stimulus turned on for a long enough duration. So the simple offset, the simple way that you defeat this as a problem is you make sure the stimulus is on for at least a half of a second. So if you do that, then you've covered the cochlear integration time, and the results that you're seeing should be valid results. Shorter than that, and or sometimes presenting at shorter durations will introduce some variability in your responses and make you more confused than you should be at that particular point. Here's just another slide that kind of shows that everything kind of coalesces. We always like slides that they all kind of come together. So all the data points as a function of frequency are all now right here at somewhere around 300 milliseconds. A little bit of a spread across frequency, but the point is as you get shorter in duration, you need greater and greater amplitudes. As we move on now to talk about differential sensitivity, slide 73 is still your slide 73. Study question 758, 759 ask about differential sensitivity. They ask sort of what is it? What are some different names for it? And so differential sensitivity, again, is the least amount of a stimulus difference you can detect. So two stimuli that differ only along one stimulus dimension, whether that be amplitude or frequency or duration. What's the least amount of a difference that you can detect? And again, the smaller the difference, the better your discriminability. Other names for this, just noticeable difference, or JND, difference Lyman, or DL, differential threshold, or just simply delta. This little triangle is read as delta. Delta S, or what's the difference in the stimulus. In general, we could think of a capacity of having the ability to hear very small differences in sounds as being equivalent to having good resolving power, a high resolving power, like you might in optics. You might think of that as a sort of high magnification. Conversely, if you can only tell a difference between two sounds that requires a very, very large difference in that stimulus dimension, that's telling us that we have very, very poor or low resolving power. Again, if you think back to one of the functions of the inner ear was to provide excellent analysis of the signal, the incoming acoustic signal, that means that we would need to have very, very good JNDs in order to be able to tell and provide to the brain excellent analysis of the amplitude, frequency, and time factors. So it would make sense that since we talked about analysis of acoustic signals as being one of the more important functions of the inner ear, it would make sense that there are some measurable psychoacoustic things that contribute to that. And that's fundamentally what the JND studies sort of are an example of. So when we think about this resolving power for amplitude, we're saying that we're able to tell small differences in amplitudes. So if we take two stimuli that are identical with the exception of their amplitude, we could tell a very, very small difference. Poor resolving power would mean that it would take very large difference in amplitudes to be able to tell that difference. When we think about, at slide 77 here, what we're thinking about, at what study question 760 is asking, well, what is the JND for amplitude? While we'll see that it's actually slightly less than this, we can characterize it sort of on an average number, so to speak, as approximately 1 decibel. So almost no matter where we are, with the exception of at threshold, with the exception of wherever we are, the ability to tell two stimuli apart when they differ only in terms of their amplitude, would be about 1 decibel. So this means if we're looking at, let's say, for example, we're looking at 1,000 hertz is the test signal, and we have it on for an adequate amount and comparable amount of duration. What that's saying to us then is that we could tell the difference between 60 dB versus 61 dB. If I played both of those two signals to you, a 1,000 hertz signal at 60 decibels and then played you a 61, and I said, are they the same or different? You'd say they're different. And the fact that you could say that they were different is because you could resolve this 1 decibel difference between those two stimuli. And that's really quite good. And so if we look at this figure from Lovin, this figure here tells us that, again, here's 1 decibel, here's 1.5, here's a 0.5 decibel. So most of our data points are actually in this region here. So I said on average 1 dB. It varies a little bit when we get near to threshold. So threshold would be this value here. This is in dBSL, where the reference is the person's threshold. So this is 40 dB above threshold. This is at threshold. So we can see a little bit of variability there at threshold, but that's not surprising based on, we know that threshold itself is a 50% response rate, so that's understandable. Once we get 10, 20 dB, 30 dB, 40 dB above threshold, we can see that we're almost all here within less than 2 decibels of this resolving power, and mostly 1 decibel all the way out. So that's just simply excellent resolving power, excellent JND. And again, we'll just say 1 decibel, even though we see it's a little bit better. Study question 761 asks about some of the implications of this excellent JND for amplitude, and these are listed for you over the following couple of slides. And fundamentally, what you have to just think about for why these JNDs are so good, or what they help us to do, is that they enable us to hear subtle differences in sounds, very small differences in sounds, and that means if we're able to hear those sounds, we're able to then decode and use very small differences in sounds as having meaning. And so therefore, when we think about this, it really has its greatest implications for our ability to understand speech sounds and to give us the capacity to process a wide range of speech sounds. Again, where is this best ability? Somewhere in the mid-frequencies, 1,000 to 5,000 hertz. Gets a little bit poorer as we got up to higher and lower frequency sounds. But basically in this mid-region where most of the understanding cues are found, that's where it is. So again, probably the biggest takeaway when we think about in practical life is that this gives us the ability to appreciate very subtle amplitude differences that might cue differences in meaning in speech sounds, or what we might say phonemic differences. At this point, we're changing the slide numbers a little bit, and so there's a bit of a difference in slide number and order here as we kind of move along, and I'll try to point out to you where the news and the olds are. So using the earlier model here just to talk about resolving power and frequency, again, what we're saying here is that good frequency resolving power means small differences in frequency, you can detect that there's a change. So the idea that I could play at 60 decibels presentation level, I could play 1,000 hertz and 1,001 hertz. You could say, and I played those two, you could say those were different. And so you could be able to resolve a 1 hertz difference between these sounds, all of the things being equal. And again, poor resolving power means you need a larger difference to be able to tell that those are in fact different sounds. So the JND in frequency tells us that if we have a large amount required to tell, that's a relatively poor frequency resolving. If you have a small difference in frequency, and you can tell that those are two different stimuli, that's relatively better. The actual amount of frequency in an absolute sense changes a little bit from 125 hertz to 8,000 hertz, the range that we're sort of thinking about as a clinical range of hearing. And the frequency discrimination threshold increases. That is to say, the amount of frequency required to tell the difference between two stimuli as a function of their frequency gets a little bit bigger as we get higher in frequency. If we, however, kind of make some generalizations here, I'm going to make a couple of generalizations for you here. If we look at frequencies that when we're at and around 1,000 hertz and we're above threshold, in that range we can detect about a 1 hertz difference. As in the example that I gave you, 1,000 hertz, 1,001, are they the same, different? You say they're different. However, as we go above 1,000 hertz, as we go to 2,000, 4,000, 8,000, we need a little bit more frequency difference to be able to tell that those two sounds are in fact different. We can get some guidance to this as we look at the next slide. And this again is looking at sort of question 762. So below 1,000 hertz, approximately 1 hertz. As we look above 1,000 hertz, so again the generalization was here. So I said it was approximately 1 hertz. Sorry to goof it up like that. And as we go up in frequency, we can see that it gets progressively more in frequency, and so on. And so at 8,000 hertz, it's bigger yet. So what it turns out to be, though, is it turns out to be that this represents a fraction of proportionality that appears to be a constant. And so as we make the stimulus difference greater and divide that by the amount of the stimulus itself, that turns out to be a proportion that seems to be a constant, and it was referred to as the Weber fraction. Your chapter in your book talks about this, and this is historically a very important early psychophysical observation that was made. And again, so really the key would be that increases in test frequency result in proportional changes in the amount of difference that's required to be able to tell. For our purposes here in COMD 4000, however, we'll just simply hang on to the idea that at around 1,000 hertz, it's approximately 1 hertz, and as we get slightly higher in frequency, it requires a little bit more. But at 1,000 hertz and below, it's about 1 hertz, and that way we've got sort of a 1 hertz, 1 dB kind of idea. And you get this proportionality thing to hold on to as well if you want. When we look at this new slide 88 was the old slide 102. And what this is basically telling us is that when we look at duration, it's difficult to assess because of the effects of frequency kind of smearing this a little bit. So there's interactions between duration and frequency. If you think about it, duration and frequency, timing and frequency are kind of interrelated. And as we already shown, there's a relationship between duration and amplitude. So this gets a little bit confusing. So we're not able to say very much, as clearly as we can with frequency and amplitude what the JND is, at least not with the same precision. Some conclusions, though, that we can make about this under ideal circumstances, that we can detect a frequency change of maybe 1 part per 1,000 for a frequency, maybe 1 part of intensity per 100 parts, and a temporal change of 1 to 10. So as we can see that, we're approximately, this is sort of a 1% over here, and now here we have like a 10% change. So from a percentage point of view, we're able to see better discriminability for frequency and intensity, and not so much for the temporal aspects. So when we think about all of these, the characteristics, intensity, frequency, duration, and we look across all of the tests that are available and all of these sort of things, intensity is the most influential, frequency the next. And our ability to tell small differences in frequency are going to vary a little bit based on where we are relative to the intensity of the stimulus. So it all kind of gets a little more complicated than we'd like it to be at this particular point. So the conclusions here, and these are the same slides, but you have these at positions 102 through 106. My 91 is your 104 or 5, in that range. our ears are well suited and are able to provide very good analysis of sounds based on the differences that occur in frequency, intensity, and duration. So that's really, really good. And our actual measured discrimination ability for these sounds really varies depending on those other acoustic conditions as well as the listening conditions themselves. But in the main, it's actually pretty good. So we can further conclude that the auditory system has excellent discrimination ability for very small changes in intensity and frequency, and less so for changes in duration. So those are our sort of takeaway implications of this. Now the remaining slides here in this slide set, that as this slide set were to continue, if you were to watch the rest of this, is where the rest of the slides from approximately 86 through 101 were. And so those are good slides, but no study guide questions exactly align with those. So on that note, we're going to say we're done with the study questions for Part 4, and we will adjourn here at Part 4